Today we will be discussing the posterior column sensations, their pathway, processing, and representation. Here I have given you a cartoon showing the caricature of sensory representation, how it looks in the brain. This is the imaginary picture, how the homunculus, the representation in the sensory cortex, how a man is looking at. Okay, I will come to that in my discussion during the course. What is special about posterior column sensation? So that means there are two sensations are categorized according to the two columns. One, the posterior column, and the other interolateral column. You know, in the spinal cord, the posterior column and the interolateral column. So these two are the columns where sensation or sensory inputs to the brain will reach. They are the fibers from the spinal cord reach to the medulla and then to the thalamus and then to the cerebral cortex. So in that we have the posterior column today. What is special about it? These are evolved sensations, what is called neo-sensations. These sensations are more precise, more accurate, more adequately dealt in the cerebral cortex. They do not cross at the segmental level. That is one aspect about the sensations. That means they come in the spinal cord, that whatever from the hand, the fibers come, reach the spinal cord, ascend up. They do not cross, they will not cross. Usually there is a crossing of sensation at segmental level or at the medulla level. So they will not cross at the segmental level. They will ascend up to the medulla, and then in the medulla they will relay, then cross to the opposite side. So that is the second part of it. And in the medulla, again relay in nuclei, which are known as a nuclei or nucleus gracilis and the nucleus cuneatus. I will talk about them. Then from these, the second order neurons start. The axons from these things cross to the opposite side and ascend as a medial lemniscus. Medial lemniscus is a bundle situated medially. Medial ribbon. Lemniscus is for a bundle or a ribbon. So ascend as a medial lemniscus to reach the thalamus, the ventero-posterolateral nucleus of the thalamus. Because they are ascending as a medial lemniscus, they are also known as a lemniscal sensation. Posterior column sensations, lemniscal sensations, both are same, are medial lemniscal sensations. Maybe you can use the medial lemniscal is more specific term. Third order neurons from the ventero-posterolateral nucleus of thalamus are projected to sensory cortex, where they are analyzed precisely, where there is a topographical representation. The distribution in the cortex according to the order of priority and the functionality. So that is the specialty of the posterior column sensations. Now, what are those sensations? Precisely, the question is, what are the sensations carried by the posterior column? That is the bundle, the posterior bundle. So what are the sensations? So these sensations, as I already mentioned, these are neo-sensations. Fine touch, tactile localization, that means identifying the exact position of the on the skin. Tactile discrimination, that is 2-point discrimination or the tactile acuity. The pressure, this is not a crude pressure, it is a fine pressure. The vibration sensation, the Steriognosis. Steriognosis is the attributes of so many components of a particular element or particular object. Steriognosis. Then the proprioception. The proprioception is carried by two set of fibers. One is a conscious sensation, conscious proprioception. Another is a unconscious proprioception. Unconscious proprioception reaches the cerebellum because it is handled by the cerebellum for immediate correction. The conscious sensation ascends through the posterior column, reach the cerebral cortex for helping the motor cortex. Again, I repeat, fine touch, tactile localization, tactile discrimination, pressure, vibration, stereognosis, proprioception. It should come. There are 7 sensations. Now, what are the differences between the touch, pressure, and vibration? That means there are 3 sensations. If you are looking at here, there are 3 sensations. This is one sensation. That is a pressure is another sensation. Vibration is another sensation. These are all the stereognosis is a composite sensation or a synthesized sensation. So what are the differences between each of them? Touch sensation results from the stimulation of the tactile receptors, which are situated immediately underneath the skin. The pressure sensation generally results from a deformation of receptors in the deeper tissues. So that means at the subdermis or subcutaneous level, it's below dermis level. The vibration sensation, if you are looking at the 3rd order of sensation, vibration sensation is a dual sensation of touch and pressure. So that means touch and pressure are alternating. So that means the receptors, touch receptors are activated once, then followed by pressure receptors, so on. This alternating the touch and the pressure that would give that sensation of vibration. So that means it has a dual receptors and a dual connections. What are the receptors involved in touch? One, Meissner's corpuscle. The Meissner corpuscle here, this is a Meissner's corpuscle. So you see that it's an encapsulated receptor. You have these nerve endings inside. This is the encapsulation. Or Merkel discs. That is the Merkel disc or the expanded cup-like structures, or egodome receptors present in the protrusions, just like the lips, the tip of the fingers, or wherever there is a protrusion, these are egodome. There is a collection of Merkel discs for one single axon. Then we have Ruffini end organs. These are Ruffini end organs. These are again carried. They are in the deeper structures. The Pacinian corpuscle. This is Pacinian corpuscle. So they are located the subdermal level. They are maybe deeper. Superficially, they are fewer. The hair end organs. So you have this hair follicle. So these nerve fibers entwine the hair, so that they will use the hair as a post to get the leverage and mechanical advantage, and the free nerve endings. So that means a touch is carried by so many receptors. There is no single receptor for touch. So because a touch is different, some touch feels the texture, some touch feels the fineness, some touch feels about the other attributes, the pressure attributes or other attributes. So all these things are managed by these 7 group of receptors present in the skin, dermis, epidermis, and the subcutaneous deeper tissues. Now, if you are looking at that, these are the free nerve endings. You just see that the peripheral nerve bundle. Here it is connected to a Pacinian corpuscle. Here, one of the touch receptor and another touch receptor. You see that entwining with the hair follicle. So that is the hair end organ. This is the free nerve ending with the hair follicle. So these are free nerve endings. These are one type of touch receptors. They are mostly seen in hairy skin. These type of receptors in the glabrous skin, that means non-hairy, especially the sole and the palm are the glabrous skin. In these things, you see there is one free nerve ending. You have the free nerve endings there. Then you have these Meissner corpuscles. The Meissner corpuscles, subdermis. This is dermis, epidermis. This is the epidermis, and this is dermis, and below the dermis level. So this. Then you have these Merkel discs. You just see that a single axon coming up. This is the egodome. This is egodome, dome-like structure. Egodome having these Merkel discs. Merkel discs. This is the egodome receptor. Then we have these, again, these are Meissner's corpuscles. So these are Ruffini and organs. These are Ruffini and organs. These are Pacinian corpuscles. So this is how the distribution of the various receptors under the skin, the dermis and the epidermis, and their association with the hair follicle, hairy areas and non-hairy areas. How these receptors behave, or how these receptors identify. You just see here the Meissner's corpuscles, and these are the receptors, the Meissner's corpuscles. You see that this is the area of representation by the Meissner's corpuscle. That means this is more precise. You can just see that this is centered here. When they are activated, when they are stimulated, this is the stimulus here. When they are stimulated, they give a burst of activity and then a silence. So that means these are rapidly adapting receptors. This is one property of these Meissner's corpuscles. So that means you can just see that they are used for texture, identifying the texture. And the hair follicle movements are used for identifying the movement. So now you have these Merkel discs here. You just see that Merkel disc. This is not as precise. See, it is more precise here, more focused. It is less focused, maybe little diffuse. And these are the egodomes. They are present in those egodomes or separately also. And their discharge pattern, when they are stimulated initially burst of activity comes, then followed by the activity continues. So that means they are slowly adapting receptors. So now we have another touch receptor here, Pacinian corpuscles. So these Pacinian corpuscles, you just look at that, it is so diffuse. The redness, you can just see that the density of those redness. So the redness only here this little bit more, and otherwise it is so diffuse. So this is a diffuse, very rapidly adapting receptors. You just see here Meissner's is rapidly adapting, and this adapts immediately, maybe within 1/100th of a second. That is the Pacinian corpuscle. These are Ruffini endings. They are situated still deeper in the deeper tissues. Again, they carry the diffuse touch sensation. So that means I have shown you, and these are not slowly adapting. These are slowly adapting. So I have two pairs which are rapidly adapting, the Meissner's corpuscles and Pacinian corpuscle, and the Merkel disc and Ruffini and organs. They are slowly adapting receptors. But the Meissner's corpuscle is more precise. You want to have the accurate information. The Merkel discs, they will stay there for long, and these are diffuse sensations. Now, what I have made in this table, I have taken this table from Genong. this table, it is about the receptor, the type of sensation the receptor carries, then adaptation, then what is the fiber or the sensory fiber associated with it, and whether they detect the vibration or where they are located. Meissner's corpuscles, they detect a texture, sharp tactile localization. I have shown that. Sharp tactile localization. And these are rapidly adapting in seconds. And they are carried by the A beta fibers. Their conduction velocity is around 50 meters per second, plus minus 20 you can add. 50 meters per second is the conduction velocity. Yes, they detect the vibration of lower frequency, 10 to 40 hertz vibration frequency. So that means they will also act as a touch receptor in the vibration receptors. They are present in a non-hairy skins, the fingertips, the lips, and genitals and other areas. So then we have the Merkel discs. These Merkel discs, they will touch edge in indentation. Say, for example, if there is a pressure here, something is pressing down, that touch, that particular depression, they will detect. And these receptors are slowly adapting. I have already shown about that. And they are also carried by the same type of fibers, A beta fibers. These A beta fibers have a conduction velocity of 50 meters per second, plus or minus 20. And they will be not carrying this vibration sense. There is a continuous sensation, so no vibration, and they are present in the hairy part. Egodome receptors are a collection of Merkel discs with a single A beta fiber. These are again a touch, and they detect the texture of the particular object. Touch as well as the texture of the object. These are slowly adapting, carried by A beta fibers with a 50 meters per second conduction velocity. They are constantly sending the impulses, and they are present in the elevated or the protruded regions of the skin. The elevated regions are the lips, the tip of the nose or the fingertips, the other areas in the body, just like a nipple in the breast or a genitals. Now, moving on to the other Ruffini end organs. These Ruffini end organs, they carry the touch. The modality of the touch is a stretch, traction. That means pulling. That type of touch it carries. And also they carry the pressure. So that means they carry touch. The nature of the touch is a stretch or a traction, and the pressure. They are slowly adapting, carried by A beta fibers. They continuously send, they are not for the vibration. So they are mostly present in the in and around the joint areas, joint capsules. They mostly detect the proprioceptive sensations because traction and the pressure. Now we have another rapidly adapting genial corpuscles. It carries a touch, it carries a transient pressure, very transient pressure, and it also carries vibration. So now this touch, pressure, transient. Here the Ruffini end organs pressure, it's continuous. The transient, so it will only small change in pressure, it only detects change in pressure and vibration. These are rapidly adapting. They adapt within 1/100th of a second, 0.01 seconds. That means fast. And A beta fibers, and these are vibration. They are at a higher frequency, 60 to 300 hertz. If you are looking at the Meissner's corpuscles, they have a lower frequency, 10 to 40 hertz, and these ones having a 60 to 300 hertz frequency. That means these are the two receptors which detect the vibrations, the Pacinian corpuscles and the Meissner's corpuscles. The Pacinian corpuscles for the higher frequency, the Meissner's corpuscles for the lower frequency, okay, of vibration. Under the skin and the deep fascia, they are present under the skin and the deep fascia. Then hair end organ. Hair end organs, they detect the touch, especially the touch in relation to the movement. In relation to the movement, if some insect is crawling, you have that particular sensation, that movement, or wind is blowing, so you feel the particular sensation. So that type of sensation, the touch as well as movement, rapidly adapting. They also adapt rapidly, but not as rapid as the Pacinian corpuscles. They may be taking about a second. A delta fibers, but these are carried by slow conducting fibers. These are fast conducting fibers. 50 meters per second is their conduction velocity. If you look at that, beta to delta, delta is half, nearly half that, maybe 20 plus or minus 10. 20 plus or minus 10 is the conduction velocity. They will be having that creeping sensation, very, very peculiar sensation. They carry that, and as if some insect is crawling in the skin. These are present in the hairy skin because hair end organ. The free nerve endings, they are located everywhere, and they detect touch and pressure. These are slowly adapting. These are slowly adapting. They are carried by the A delta fibers. They do not take the vibration, and they are everywhere. Tickling sensation comes from these free nerve endings. So this is about the receptors and the modality of the sensation these receptors detect. I have mentioned the movement, the vibration, then attraction or stretch. I have mentioned about the sharpness and the localization, indentation, texture. These are the characteristics of the different type of sensory, touch sensory modality. What happens? They are carried by the A beta or A delta fibers. This nerve divides into two branches, one medial, another lateral. The medial branch ascends up as the posterior column, either as a fasciculus gracilis or fasciculus cuneatus. The fasciculus gracilis is the part, the lower limb gracilis is the lower limb, and cuneatus is the upper limb and the neck. So it, from the site where they are originating, they will ascend as these bundles, cuneatus or gracilis, and reach the medulla. So this is one part. So what happens to the lateral part? Because it gives another collateral. The lateral collateral, it makes a synaptic contact at 3 or more places. Okay, so the first place, it will make a synaptic contact here with the motor neuron for the reflex activity. These are flexor reflexes, the flexor reflex afferents. So the flexor reflexes may be withdrawal or anything. So that means that this is for the spinal reflexes, this part. Then they will give a collateral synaptic contact with the fibers. These fibers ascend as the spinocerebellar tract. This is a dorsal spinocerebellar tract, and this is a ventral spinocerebellar tract. These will ascend as a spine. That means they are carrying the type of position sense, the proprioception. This is the dorsal and ventral spinocerebellar. So then another part is it will be sending the impulses to the cervical tract. So that means the lower part of the vertebral or the spinal cord should have a link with the cervical portion, because when we are maintaining the body posture, body on neck, body on body, So these type of reflexes, they exist. So now here, the same thing here, the fasciculus gracilis, because it is coming from the lower limb. If you are just looking at this, is the drawing I have made and I have taken the picture here. This is coming from the foot. So as it ascends, it comes here and it tries to as from the foot the leg is coming. As the leg is coming, this part goes medially. So like that, they keeps pushing. When it comes to the hand, that is in the thorax and cervical area, so that will occupy in this part. So this is the cuneate. This would be pushed medially, and this would become laterally. We have a fasciculus gracilis from the lower limb, fasciculus cuneatus from the upper limb. So they will reach to the medulla. So in the medulla, we have the nucleus cuneatus and the nucleus gracilis in the medulla. So they don't cross, they will ascend in the posterior column. So now they will ascend here. And once they relay, the second order neurons begin from this. And these second order neurons, say for example, the nucleus gracilis, it will ascend up. So if you are looking at, there will be some configurational change in this diagram, little bit more. It's not clear. These are called arcuate fibers. These fibers, the crossing fibers, are known as arcuate fibers, and the internal ones, they become outside. The internal ones, they become outside. Here it is not represented properly, and the external ones, that is in the lateral ones, become medial. That's what I have shown here in this picture. So that means in the medulla, when crossing, the lateral one become medial and the medial one becomes lateral. So that is through this arcuate fibers, very peculiar arrangement. And then this one ascend as a bundle, the medial lemniscal bundle, or it's also known as a lemniscal. Lemniscus is a ribbon. It's a ribbon-like structure that reaches to the thalamus. In the thalamus, we have a specified nucleus that is called ventro-posterior lateral nucleus, VPL, the ventro-posterior lateral nucleus of the thalamus. That is where it relays. And from the ventro-posterior lateral nucleus of the thalamus, it will give to the cortex. This is a parietal cortex. This is the parietal cortex. That is the pathway for the posterior column sensations. Here, I have made the somatotopic localization at each level. At each level, there is a somatotopic localization. So you just see that S is for a sacral segment. They are most medial, then lumbar, then thoracic, then cervical. So like that, the newer one in the spinal cord, that is the arrangement. The cervical ones are lateral, and the sacral ones are the most medial. So if you are looking at this thing, okay, what I did mention here, this fasciculus gracilis is known as a tract of gall, and this fasciculus cuneatus, a tract of Burdak, with the name of the scientists. Now, going to the further, I am just trying to make it in the words here. So, pathway for posterior sensations. That starts with a receptor. I have mentioned the receptors, the Meissner corpuscles, the Pacinian corpuscles, the Merkel disc, Ruffini end organs, free nerve endings, egodome receptors, and the hair follicle receptors. These receptors are carried through the fibers A beta and A delta and reach the spinal cord. This is number 2 step. So they are coming here, they reach the spinal cord, and then they ascend as fasciculus gracilis or fasciculus cuneatus to the medulla. In the medulla, you have the gracilis nucleus and the cuneate nucleus, where they relay in the nucleus gracilis and nucleus cuneatus, and then cross over to the other side other side, and then ascend to thalamus as a second order neuron, as a medial lemniscal bundle. And when they are crossing in the medulla, they form the arcuate, the internal arcuate. So that is the curved position. Okay. So then in the thalamus, they relay in the ventro-posterolateral nucleus of thalamus, and then they are third order neurons relay in the parietal cortex area, Brodmann area 312. 1 to 7 are the pathway for posterior column sensations. You want to remember them, where these fibers are projected. Now I have just drawn the diagram. So this is the brain here, this is the brain structure, this is the frontal area, this is the occipital area, this is the temporal area, and this is the parietal area. This is the central sulcus, this is central sulcus, this is lateral sulcus. So now this area, what I have made with the blue pen dotted line, this is the somatosensory area. And the somatosensory area, you just look at that 3, 1, 2. I never said it is 1, 2, 3. 3, 1, 2. 3 is the interior portion of this sulcus. 1 is on the top of the gyrus, and 2 is another side of the sulcus. 3, 1, 2 is the somatosensory area number 1. That is where the first neurons are relayed. And here there is a topographical representation of each and every part of the body here. So now from here, the attributes of these sensations are processed with the help of somatosensory area 2 and posterior parietal somatosensory association. This 5 and 7 are the association areas, the posterior parietal association areas, and S2 is the assistant, where the attributes of S1, the touch, are deciphered or decoded. So this is what this posterior parietal area is in connection with the visual area. Occipital area is the visual area. It is in connection with the visual area, and this is also in the posterior parietal area is also in connection with the auditory area, and this is also in connection with the here is the limbic structure. It is also connection with the limbic area. So what does it mean? The visual area, the auditory area, and the limbic area. Okay, so that is what I have mentioned here. This is where it is projected. Now let us see the diagram, real diagram. So you just see that I have, see here intentionally I have drawn with my hand, because I could have loved to draw it in a chalkboard there, so that you will be able to draw it in your own this thing. So now here the same thing. This is occipital, this is frontal, this is temporal, this is parietal, this is central sulcus, this is lateral sulcus. Now this blue one is the somatosensory area 1, and this one is the somatosensory area 2. Here is the 5 and 7, the somatosensory association area. So that is what the representation is. If we can take the section, the coronal section, it's a coronal section, we are cutting it like this. So then what happens? This is one half, that is another half. The interior part, so say for example, here if you are looking back in the interior, it will go down, it is carved like this thing. This is the lateral part. The interior part here is the representation for the genitals. This is the little greater representation is there for genitals. So then toes, then the foot and the leg, very little, hip little, hip and buttock region very little, the trunk very little, and the neck, head, the shoulder, and the arm, elbow, very little these things. Forearm, it is becoming a little longer, wrist is more, the hand is a large area, and again the fingers, each finger have a area for representation. The little finger, ring finger, middle finger, index finger, thumb. Thumb has a largest distribution. Okay, so then eyes have a representation because we have to have the sensations coming from the eye. Then the nose, then face, the upper lip. See, this is the type of representation. The greater face is having a greater representation. You just see that face. The lips, they are very large area, lower lip. So then teeth, you have relatively large area, tongue, large area, the pharynx, yes, it is sensitive. The entire intra-abdominal visceral organs are represented laterally. Very little is there because so many viscera are there. So this is what is called a homunculus, sensory homunculus. So now, to summarize this, what is this sensory homunculus is about? There is a greater area of representation for hand. You can just see that. The hand is large. Fingers, face, lips, and tongue. You all agree with me. Hand is large. The face is large. Lips are thick. And the tongue. Little for the trunk and hip. Very little for the trunk and hip. So you just, if you are looking at the body size, there is a large part is the trunk. But the sensations carried by the trunk are not much. And hip are not much. So whereas the sensations carried by the fingers and the face are large. So that is where the importance is. The least for the viscera. Area of the cortex assigned for a particular part is proportional to its functional significance. That is what. So that means the greater the function, the greater the representation in the cortex. If there is no function, the less. So you say, for example, here, the trunk, if you are looking at, or the knee you are looking at, or the buttocks you are looking at, you have a very little representation. Thumb, fingers, lips, tongue have a disproportionately large representation. That is what makes the sensory unculus. I want to impress upon you that disproportionality is due to the functionality of the particular part or the sensory functionality of the particular part. So now, looking at that, suppose if I extrapolate this all features and I can construct a caricature. So what this caricature looks like? Very large face and a very large tongue. Eyes are protruding. Lips are thick. See, look at the lips are thick. And so you see even the ears are more, but neck is very little, and even the thorax maybe even lesser than what they have represented. Thorax is lesser. The entire trunk is only a small bit, and the arms may be still thinner, and the hands are big. That is okay. You just see that such a large hands, and even the legs may be a little thicker. No, it's not required that much. It is thin. It should be thin. All these things should be things. But whereas genitals have a greater representation, so that means a larger genitals. So that means that's what I have mentioned: the large face, big nose and eyes and pinna, thick lips, large tongue, very big hand and the fingers. Thumb, fingers, lips have a disproportionately large representation, slightly bigger genitalia, and remaining parts are thin and emaciated. This is a sort of a caricature, a monster-like look. So that is what the homunculus of the sensory homunculus. Now coming back, so relay in the cortex and processing. So now what happens in the cortex? We have seen that it is relayed in the cortex. It does not mean anything. You have got the Xn potentials arriving in the cerebral cortex. Where in the cerebral cortex? The area located in the parietal cortex is in the posterior central sulcus. It is known as a primary sensory area, or Brodmann classified them as areas 3, 1, and 2, and it is also known as a S1, somato-sensory area 1. The topographic representation of various parts of the body are shown in the homunculus and is proportional to the degree of sensory function performed by that organ. Thalamocortical projections relay in the inner granular, where they relay in the cerebral cortex. The cerebral cortex is divided into 6 layers. And in that 6th layer, layer 4 is a granular layer. And in this layer, the thalamic outputs, they come and relay. That is in the layer 4 of the Brodmann area chamber. So topographical localization and sensory modality is, so now if I stimulate the particular area in the cerebral cortex, that would produce the sensation of a touch in the finger or whatever. Suppose if this finger, if I were to, I should go to that particular site and stimulate, that would produce the touch sensation. That means there is a topographical localization. S1 gives outputs. Now, S1 has only identified the arrival of the impulse. That means it identifies the location of the impulse. Yes, it is located in the arm, in the finger. But the attributes or the fineness of the touch are to be processed with the help of S2 and somato-sensory association areas. Because in these parts, the processing is done and attributes are given. So say, for example, you take any objects, each object has certain physical parameters, and whether it is a metallic, non-metallic, whether it's a plastic, wooden, or whether it is warm or cold. So each of these attributes are processed in the cerebral cortex in association with these S2 and somatosensory association. So that means the lesions of this primary sensory area so lead to the loss of appreciation of the exteroceptive and proprioceptive sensations from the opposite half of the body. So that means if there is a lesion of the left cerebral cortex, the parietal area, left parietal area, so that means the right side sensation is lost. Right side sensation of that part is lost, because the parietal area is quite large. So now coming back, so here receives impulses for touch. This one, the primary sensory area, receives impulses for touch, pressure, vibration, itch, tickle, proprioception, joint and muscle position. Involve perception of these sensations. They involve the perception of these sensations, allows you to pinpoint where exactly somatic sensations originate. You can pinpoint it where it is originating. So that is why you can know that if a mosquito is biting here, and you know that you can hit there only in that particular position. So that means that is the tactile localization, tactile identification. That means you identify the particular point, pinpoint these things. That is what I mentioned here. You know exactly where to hit the mosquito. Now, what are the functions of the somatosensory area 2? The somatosensory area 2 is located in the upper lip of the posterior image of the lateral sulcus. See, for example, this is lateral sulcus. This is lateral sulcus. This is the upper portion of the lateral sulcus. So that's what exactly is mentioned. Upper lip of the posterior ramus of the lateral sulcus. This is also known as the Sylvian fissure. These areas are represented by Brodmann as 40, Brodmann's area 40 and 43. These areas receive inputs from S1. So again, if you are looking at the face area is here and the leg area is in the backwards. So that face area is mostly anterior and the leg area is posterior, backwards. Whole body is represented bilaterally. The output from S2 will go to posterior parietal area, 5 and 7, and the limbic association area. So the 5 and 7 are here. This is 5 and 7 here, and the limbic association area here. So the output from these S2 will go to the 5 and 7, that is the association area and limbic association area. So now, what are the functions? It is concerned with the tactile discrimination, to discriminate 2 points: the spatial location where exactly is situated, and other information or attributes of the objects stored in memory. So whether it is a rubber or a plastic, wooden, metallic, or all those characteristics or attributes are stored here in the memory. That is about S2. So that means if the S2 is lost, so these attributes are not. So he may be able to locate the sensation, but if he will not be able to say that what this object is made up of, he cannot be able to tell whether this object is made up of a plastic or a metal or a wooden or a pot or something like that. So he will not be able to do that. So then we have a third area. This is the posterior parietal area. It is also known as somato-sensory association area. This is the somato-sensory association area. This is S1. This is S2. This is somato-sensory association area, the posterior parietal area. So now it is located superior posterior lobe of the parietal cortex. So this is the parietal cortex in the superior part, posterior. Okay, concerned with the perception of the shape, size, roughness, texture, orientation, and direction of motion, angulation. So that means it has a large group of attributes which it has to analyze. So that means anything about the objects, it does. It does. Now, in this area, all our sensory modalities are stored in memory. The areas, these are association areas. So I remember whatever the objects I will take. Say, for example, if I take the pen, so I remember about the properties or attributes of the pen. So then this pen is a plastic one, that is remembered there. So then when I touch it, it is a plastic pen. I know that because of the attributes, it is a pen. Otherwise, it's a rod or a cylindrical structure. So now coming back here, other details of the objects in reference to the sensory modalities are analyzed and decoded here in this area. And in addition, this part is in contact with the visual association area, auditory association area, and the limbic association area. This is visual association area here, auditory area here, and the limbic association area. Visual association area is for object imagery. Say, for example, I am holding this pen. Even if I close my eyes, I can have an image of that particular object. So that is because of the visual association area. And it is a type of, I can very well assess if it falls, what type of sound it may produce. That object, the property, if it is striking to some other part, ground. So that the auditory part, it may not make a metallic sound, it will make a flat sound. So that's what I can talk about. So now, suppose it is also concerned with the now emotional components. Emotional components, it is a pen. Say, for example, even whenever we worship it as the goddess of learning, Ma Saraswati. So now we do not want to, suppose if I just try to touch with my foot, I immediately have that emotional component attached. Emotional components, I take it in my hand and I pray the God. So that's what we all have been doing it, and hope you are also doing it. That's an emotional component. This is about a pen. Suppose if I have given you the knife, The emotional component of the knife, it is going to harm. It may produce injury. So that type of emotional component is coming with the limbic system. So that means all these attributes are deciphered here in the posterior parietal area, what's called a somato-sensory association area. So in addition to the exact attributes, the perception, size, shape, roughness, texture, orientation, direction of motion, angulation, so on and so forth. In addition, their visual imagery, auditory associations, and the limbic associations, all those things are attributed. And this is the area which is necessary for stereognosis. Stereognosis is identifying the structure. Say, for example, if I close my eye and if you ask me to hold this part, I will say that it is a pen. And you will say that with these attributes, I will say that it's a ballpoint pen. With without any cap, there is a pressure. These things. So this type of this is known as a stereognosis. And without this thing, the stereognosis is not possible. In that condition is known as a astereognosis. Now, what are the various clinical abnormalities associated with the sensation? One thing is sensation is carried by the large diameter fibers. The large diameter fibers are having a myelination. So anything which is producing a demyelination, as happens in case of a vitamin B12 deficiency, what is called as a subacute combined degeneration. The vitamin B12 deficiency produces the myelination problem. So that is one. So they are affected. Or multiple sclerosis, demyelinating disorders that will affect. Or the tertiary syphilis, the tapes dorsalis, they affect these sensations. So where large diameter sensory fibers are affected, and these are the three diseases I have mentioned. Vitamin B12 deficiency, because the myelination is hampered, demyelinating disorders, that is the multiple sclerosis, and the tapes dorsalis, a tertiary syphilis. So that result in the loss of posterior column sensations. That is one aspect. So now Saturday night paralysis. Saturday night paralysis, that means the mechanical pressure blocks the conduction of impulses in the large diameter fibers. The mechanical pressure. A person after boozing in the Saturday, and he sleeps over on his arm, and then he will have the paralysis of the upper limb. So because of the large diameter fibers. So this is known as a Saturday night paralysis, or maybe the large diameter fibers are sensitive to pressure, hence the transient loss of sensation. The first degree neural injury happens, neuropraxia, and that is called the posterior column. That is posterior column. Now, lesions of the primary sensory area, the loss of sensations such as fine touch, vibration, discrimination, localization, 2-point discrimination on the opposite side of the body. These are all lost. Fine touch, the vibration, discrimination, localization in the opposite side of the body. One more word about vibration. So in diabetes mellitus or diabetic neuropathy, long-standing diabetes mellitus will have a neuropathy. And in this neuropathy, the vibration sensation is the first one to be lost. Even in these conditions, where the subacute combined degeneration or demyelinating disorders, the vibration sense is the first one to be lost, because the vibration is carried by a dual fibers, so that is it's more vulnerable. It's more vulnerable. So that is one part of the loss of sensation. I mentioned about the astereognosis, loss of stereognosis because of the somato-sensory association area is lost. Inability to recognize or identify the commonly used objects. Just like a coin, I can identify the coin denomination of the coin, and so on. Any objects you give. Ideomotor apraxia, that is, lesions of the left superior parietal lobe, are associated with the loss of ability to produce purposeful, skilled movements as a result of a brain pathology not used by weakness or paralysis or coordination or sensory loss. Posterior column sensations are neosensations. They are fine touch, tactile localization, tactile discrimination, vibration, pressure, proprioception. Steriognosis. Receptors are Meissner's corpuscle, Merkel disc, egodome receptors, Ruffini end organs, Pacinian corpuscles, hair end organs, and free nerve endings. They are carried by the A beta and A delta fibers, the myelinated, to the spinal cord and ascend as a fasciculus gracilis and cuneatus to relay in the respective nuclei in the medulla. The second order neurons cross, the axons of the second order neurons cross and ascend as a medial lemniscal bundle to relay in the ventral-posterolateral nucleus of thalamus. Third order neurons from thalamus reach postcentral area 312 of parietal cortex, that is area S1, somatosensory area 1, where they are represented topographically, pinpoint localization. Area of representation is proportional to the functional importance of the sensation or functionality of the sensation. Then S1 area in association with the S2 and sensory association area decodes all the attributes of the sensory modality and brings the total picture of the sensation. That's the summary. This I will refer to the guidance textbook of medical physiology, Ganong's review of medical physiology, and one or two graphs are taken from the Candle's textbook of neuroscience and Samson Wright's physiology. Okay, so now the assignments I have, because you just see the number of questions they are coming up, because these are the questions you have seen them, I have seen them in your universities, these things. Here I name, I think it is for your working, name the various medial lemniscal sensations or posterior column sensations. Describe the pathway and processing at various levels. I have dealt in a comprehensive manner. Draw a neat label diagram showing the pathway for tactile localization and tactile discrimination. So that means you have to use that the Meissner's Merkel discs and discrimination. So they describe briefly the processing of these sensations. Again, the answer is same. Describe the receptors, pathway, processing of vibration sensation. So that means the receptors are Meissner's corpuscles for lower frequency and the Pacinian corpuscles for higher frequency, and how they are carried and what are the fibers where they reach. Explain the phenomena of early loss of vibration sense in diabetic neuropathy, because it's a dual pathway. Then the short notes on vibration sense, the posterior column sensations, Saturday night paralysis, the diagram, write a diagram showing the various areas handling the somaesthetic sensations. That means you try to draw the cortex, S1, S2, and somatosensory association area in the cerebral cortex. Astereognosis, 2-point discrimination. Somatosensory association area 1. So then you have to give all the functions and representation. Somatosensory association area 2. And posterior parietal association area. The sensory homunculus, I have not included, maybe you may have to. The tactile localization, sensory receptors for touch. Each one may find a short note question. Merkel disc, Meissner corpuscle, Pacinian corpuscle, Ruffini and organs. I have dealt with them in a little detailed manner in my discussion. Okay. In the next class, I will deal with the organization of the cerebral cortex before I take other sensory modality. Thank you.